All right, so thank you, Ms. Khalid, for the time. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good day, everyone, and hopefully you all have, ladies and gentlemen, here have a very wonderful day today. And for the first session of our class of today, it will be delivered by Miss Shofa Nabilah in the topic of the genetic engineering in animals. And I'm here, Muhammad Novana Shabana, as your moderator for today. And without further ado, I will give the time to Miss Shofa Nabilah. Miss Shofa Nabilah, are you ready?
Yes.
Okay, then go ahead. The stage is yours.
Okay. Thank you, Nabilah. Now let me share the screen first. Okay. Hello, everyone. Good afternoon.
Good afternoon.
Okay. So I will explain about genetic engineering in animals. So what is genetic engineering in animals? So the definition is manipulation of genetic material. So genetic engineering is the manipulation of genetic material that is DNA or RNA. So the objective of bringing about the innovation and it is carried out during this study or modification for any purpose. So the types of genetic engineering in animals, the first is xenografting. So xenografting, the process is transferring tissues or whole organ from animals donors. So the first case of xenografting carried out in 1984. So the case is baby pig who have disease like have a hole in their heart. So he get a transfer organ from baboon's heart. So a baby pig can live with baboon hearts for 3 weeks, and after 3 weeks, she died because the human immunity rejected the baboon's heart. And also xenografting example is also a pig organ. So the researcher still research about the pig organ that can transfer the cell or the tissue or the organ that can be used to human. And the second is gene manipulation. So the process is isolation of genetic material from an organism and transfer of genetic material to a sustainable vector, and then transferring of a vector to a host through transformation. So in here, gene manipulation, the example of gene manipulation is pig that can produce human insulin that can be used to treat diabetes. So many example that we'll explain in applied genetic engineering in animals. The first is companion animals. So the companion animals example is goldfish, and here are mice, and it's cat. Sorry, it's not quite clear. I did zoom in. So why it so glowfish and other animals, it can be glow in the dark. So why it can be like this? Because the scientists have injection their genes from any anemones and jellyfish. jellyfish. So the code for that makes the animals can glow, it's because about fluorescent proteins that get from what jellyfish. And genetic engineering in animals, example is wild animals. So researchers still plans to clone the extract tilasin. Here is like what Strigala, what English I forget, plans to clone the extract and the woolly mammoths. So it's still research, not still research by researchers. And from animals also apply in genetic engineering. The example is pig that have higher level of omega-3 and goats that have human lysozyme in their milk. So genetically engineering from animals can be created to enhance the quality of the products, the food. So pigs have been genetically engineered to express the D12 fatty acid desaturase gene from spinach, and for higher level of omega-3. And goats have been genetically engineered to express human lysozyme in their milk. And also, genetic engineering in animals can be used for food production and healthcare. For food production, that has been explained before is farm animals that purpose for high food quality. And for healthcare, the example is cancer research and human gene therapies. And scientists have been major breakthroughs in organ transplantation, cancer research, and other areas. Similarities between the genome of human and other animals also suggest that future genetic research on animals will yield additional benefit for human. And animal genetic engineering can also help in developing human gene therapies. In one experiment, scientists have used genetic gene therapy to 70% of mice implant with one kind of human ovarian cancer. So I think this is enough for the presentation.
Okay, thank you very much from Ms. Sofa Nabilah for the great explanation about the genetic engineering in animals. And now, after the presentation has finished, we are opening the Q&A session. The Q&A session will be divided into two sessions. The first session is going to be open for 2 questions, and the last session will be dedicated for the three questions. Okay, so without further ado, does anyone who want to ask a question or to discuss something? Okay, we have Agnya and Mutiara. Okay, so Agnya, go ahead. You want to ask a question? By the way.
Okay, first of all, thank you, Naufal, and thank you, Shofa, for presenting such amazing material. I want to ask, are all of them successful experiment, or is there any failed experiment on genetic engineering? If there is one, could you please tell me? Thank you.
Okay, thank you, Ms. Agnya, for the question. So, Ms. Sofa, will the question be collected first, or you will direct answer the question?
I think collected first.
Okay then. So we're gonna collect the question first. And now we're moving on to Miss Balkis. Go ahead.
Thank you, Kang Iweng. I want to ask, is genetic genetic engineering in animal same with cloning animal?
Okay, Miss Balkis, thank you for the question. So do you need time to answer the question, Miss Shofa?
Yes. Wait.
All right then. All right, we will wait for Miss Shofa to get the answer first, and we'll be right back.
Okay. I think I will answer the question from Teh Agi. Teh Aya. Okay. So the genetic engineering in animals, the successful experiment is what is example is here, goldfish and other animal like glow in the dark. And from animals that pig have a higher level of omega-3 and goat that have human lysozyme in their milk. And that not successful is about xenografting, because xenografting what regarding to what mencocokkan organ from animals and human, right? So it's still developing. So I think not filed, but still develop and still apa? Still tetap dipelajari gitu. Gimana cara yang baiknya agar mendapatkan hasil yang sesuai dengan penelitian. Mungkin itu.
Thank you, Miss Shofa.
Okay. Okay, so for the next question about what same cloning animals and genetic engineering in animals, right? So cloning produce genetically exact copies of organism. So the organism is like what? Copy and paste. But genetic engineering in here refers to process in which scientists manipulate the genes to create new products. So many manipulation in the products that can the products. So different version of organism. So cloning is like copy paste. That genetic engineering is manipulation, like that.
Okay, so that will be all of the question has been answered. All right, so that is for the first session of the Q&A. And now we will open the second session of the Q&A. All right, so if anyone, one of you who want to ask a question, will go right ahead. Okay, there's two. Okay. Okay, so there's still one slot left open. So if you want to ask question, go right away. And now we have Miss Riza who want to ask a question. Go ahead, Miss Riza.
Okay, thank you, Sir Novel, for the time, and also thank you so much for Miss Sofa for the very spectacular presentation. However, I still wonder about something, so I just briefly read from the Federal Agency of Food and Drug Administration from USA that it is stated that for food production out of the genetic engineering animals, it is not allowed, or it is like forbidden, and it's like against the rules. So I wonder, in your, what is it, the mind map, why is the use of food production is allowed? Is the regulation different between every countries, or can we just go against the rules, or what? Because it is stated by the FDA itself. And yeah, that's my question. Thank you so much.
All right, thank you, Miss Riza, for the question. So it will be collected again. Yes, Miss Sofa.
Yes.
Okay, then. All right, let's move on to the next one. It will be Miss Arlieza. Go ahead, Miss Arlieza.
Okay, thank you so much, Sir Novel, for the opportunity. And thank you, Miss Sofa, for your great presentation. Actually, you've already delivered about the advantages of genetic engineering in animals that have been applied in our life. Then I'm curious, how about the risk or disadvantages of doing genetic engineering animals in our life? So that's the question. Thank you.
All right, thank you for the question, Miss Fazla. Okay, so for this one, Miss Shofa, do you need time to answer the question?
Yes. Wait a minute.
All right then. So while we're waiting, we still have one slot for the question. So any one of you who want to ask a question, feel free to ask.
Okay, I think I will answer from the early Zafazla question. So the risk or disadvantages for genetic engineering in animals, for example, can be toxic from the crop have been detected in some people's blood. So potential harm for human health is like what antibiotic resistant and concentration of toxic metals. Still what have disadvantages from genetic engineering in animals, and also yes, I think it's the answer.
Okay, thank you so much, Miss Sofa, for your answer.
So I will explain answer from Tehriza's question. The food that creates by genetic engineering is allowed or not allowed, yeah, to the question. And the risk of food from genetic engineering, it's also have what disadvantages from some people, that some people have new allergy in from this food, and some of them is not have any what have any negative sides for the food that creates by genetic engineering. But I think it's allowed, because many products that sell in other countries, but in Indonesia, I think it's not like common. It's not common food that created by genetic engineering in animals, but in other countries is allowed.
Okay, thank you, Miss Sofa, for the answer.
Okay.
Okay, so that's it for the question. I would like to remind you again, ladies and gentlemen, there is still one slot open for the question session. So if you have anything in mind, if you want to ask, go right away, because we still have one slot left. Okay, I see. If there is no one, I will count down, starting from 5. So to end this Q&A session, 5, 4, 3, 2, 1. Okay, so that's it for the second session, because there are no ones want to ask the question again. And now we are at the end of the first session of our class today. Okay, so before we get to the end, let me read the conclusion for this the first session. So the genetic engineering in animal itself, it's mean that the manipulation of the genetic materials in the animal itself. It could be it divided into two types. The first is xenografting and the gene manipulating. It has very applied, which could be applied to your pets, to the wild animal, and to the farm animal. And for the use of itself, it could be used in the healthcare field and also in the food production. And that's it for the conclusion of today's session, the first today's session. Thank you very much for your attention. And thank you, Miss Sofa, also for the amazing presentation. I'm your moderator for today, Muhammad Naufal Nur Syabana. And yeah, see you when I see you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.